As you are walking in the real world, everything is happening. And these are massively complicated calculations, and in virtual reality, these laws are being broken. Never before in history of animals, two senses said one thing is happening and two other senses said, nope, that's not happening.
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 191st episode of this podcast dedicated to anything and everything that you can do to help the better workings of your own brain, all of which becomes easier if you actually know where you are. Which raises the question, where are you exactly? Something which you almost certainly know the answer to, but you know that answer because of something in your brain called place cells, which help you to constantly check and double check your environment, making sure you are where you seem to be, you're aware of it if you get lost, and of course can be laying down memories of new places. Humans are not alone in having this sort of neural hardware. Obviously, this is something that comes in extremely handy in the animal kingdom, and it's also something that is tripped up in interesting and not immediately apparent ways by virtual reality. We're going to be talking with something of a polymath who has done a lot of study into place cells, spatial location, virtual reality. Dr. Mayank Mehta will be our guest in the main interview. If you hang around until the end of the episode in the Ruthless Listener Retention gimmick, I'm going to tell you about a new study exploring the misty origins of the anxiety that you might feel while parallel parking and how this may or may not have any relation to how anxious you feel while playing Jenga. But for right now, let's kick things off, as usual, with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So if any of you are competitive cards players, you know that you're supposed to be looking for tells in your opponents. A tell would be some sort of giveaway, typically physical, maybe verbal, but something where a person is giving away what's going on inside their head without having meant to and probably without even realizing that they've done it. And scientists at Ohio State University might have found a common tell, at least in the way that people operate a computer mouse, that's giving away just how tempted they are by certain temptations, even when they know that those temptations fly in the face of their current goals. This research, which is about to appear in the journal Psychological Science, involves several experiments. In one of them, 81 college students made 100 decisions involving healthy versus unhealthy food choices. The study subjects clicked a start button at the bottom of the screen, and as soon as they did, two images would appear in the upper right and upper left corners of the screen. And one of the images would be a classically healthy food, something like rutabagas or Brussels sprouts. And the other one would be something unhealthy, but probably more tempting, something like a pastry or a bag of Skittles or something like that. And the participants were told that as soon as the images appeared, to move their mouse as quickly as possible to the image of the food that would do the most to support their current health goals. So a pretty unambiguous right answer. And they were also told that at the end of the experiment, they would be able to pick a real food to eat, whatever it was they wanted. So there was a right answer to these questions. The vast majority of people wound up choosing the right answer. There weren't many people that clicked Skittles when they were saying go for the healthy thing. But whereas some people's mouse trails showed a beeline for the healthy object, there were other people that kind of made more of a curving line. Like it started going for the unhealthy thing, and then they thought better of it, and their hand arced in towards the correct answer. And looking at whether the people's mouse trails were showing straight lines for the correct answer or were taking a more arcing path to get there, the researchers could very accurately predict the real-world food choices that were made by the participants after the end of the experiment, or after the alleged end of the experiment, because part of the experiment was actually seeing what they would choose in the real world. The people with arcing paths in their mouse trails were far more likely to go for the candy bar offered at the end of the experiment than the apple, said Paul Stillman, the study's co-author. For those with lower self-control, the temptation is actually drawing their hand closer to the less healthy food choice. The more that they were pulled toward the temptation on the computer screen, the more they actually chose the temptations and failed at self-control. They also did essentially the same study with a totally non-food-based scenario where people could choose whether to accept $25 right now or $45 in 180 days. Again, there's almost certainly a right answer to this question. If you can give somebody $25 today and they'll reliably give you $45 back in 180 days, do that till you're blue in the face. But the people that were more likely to make the real-world bad decisions also tipped their hand with this arcing mouse move when the choices appeared. Something about this finding of the arcing paths, it kind of reminds me of somebody shooting a gun in high winds and needing to compensate for the wind speed. Everybody's eventually getting to the right answer, but you've got this extra force that you've got to factor in. In this case, the wind is sort of analogous to that temptation, and your executive function needs to compensate for that in order to move the mouse to the right choice, said Stillman. Our results suggest a more dynamical process in which the healthy and unhealthy choices are competing from the very beginning in our brains. There is no abrupt change in thinking, and that's why we get these curved trajectories. This mouse tracking metric could be a powerful new tool to investigate real-time conflict when people have to make decisions related to self-control.
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
Picked up a couple of 5-star reviews on iTunes. Listener Estelana from Australia says, I've been listening to SDS for at least 3 years now. With a seemingly endless line of credible experts and fascinating topics, I refuse to miss an episode. And listener Jeff Godin from the USA says, informative and concise, tons of great information in a concise manner. And a hat tip right back to Jeff for a nice, concise review. Thanks, of course, to everybody that's been leaving reviews on iTunes or otherwise spreading the word on Smart Drug Smarts. Word of mouth is, of course, incredibly valuable. We appreciate your vouching for us to your brain and science curious friends. You can pop them off a link to smartdrugsmarts.com. You can find any Smart Drug Smarts episode in our entire arsenal by going to smartdrugsmarts.com/whatever the number is. This episode will be smartdrugsmarts.com/191, as you might imagine. And of course, if you want reminders as new episodes are coming out, a lot of podcast apps will do that. If you kind of subscribe and say, hey, I really like this one, let me know. But you could also sign up for our newsletter, The Brain Breakfast, at smartdrugsmarts.com/newsletter. I've been mentioning for the last few weeks the launch list at Vitamunk. Just a couple of days ago, they sent out an email with an offer for a product. I'm not joking. It's called OptiGoat. Goat as in the barnyard animal, but this is actually a product, an extract of something called horny goatweed, which we've not done an episode on that. Might save that for a Valentine's episode next year. We'll see what happens. That one, as you might gather from the name, is more about the libido than cognition. But my point is, it was a screaming deal, under $5 for a jar of this stuff, only offered to a small number of people. They're just trying to jack up Amazon sales at the beginning as they open new product lines. But a good email list to be on, good thing to be aware of. You can sign up for that if you're curious at smartdrugsmarts.com/launch. And of course, never far from my mind or my mitochondria are Nexus and Mitogen, the couple of stacks we have over at Axon Labs. We'll be doing some sort of Dog Days of Summer event for Axon Labs in a little bit here. Keep your eyes peeled for that. And of course, you can find both Nexus and Mitogen online at axonlabs.io.
Smart Drug Smarts.
So Dr. Mayank Mehta, who I'm about to be speaking with, has one of those career resumes like a scientist from the 16th century, where somebody could be at or near the top of the game in astronomy and physics and biology and medicine all at once, which isn't something that really happens anymore. But Dr. Mehta makes a pretty good run at it. He is with the UCLA Department of Physics and Astronomy. We will not be talking about astronomy in this episode. He is also part of the Department of Biophysics, and in particular neurophysics, and looks at questions like how are representations of the physical world represented by neurons? What are the roles of the brain's rhythms, things like sleep and awake? And of course, seeing how lab rats respond to various perturbations of their senses. We're going to be talking about all of this and more, particularly in the context of something called place cells. Place cells are one of the worst named things I can think of in science. If you're naming a kid, you shouldn't name the kid something where the last letter of their first name ends with the first letter of their last name. If you name somebody like Bob Boyd, it makes it really awkward to say because you want to run it together into Bob Boyd. But unfortunately, place cells have this nasty sibilance, so I apologize if both me and the interviewee spend the whole episode sounding like we're lisping. But what they lack in pronounceability, they more than make up for in being downright interesting. But look around, get a good sense of where you're at, and prepare to have the foundations of your physical reality neurologically shaken by Dr. Mayank Mehta.
I am Mayank Mehta, professor in the Department of Physics and Astronomy and Neurology and Neurobiology. So you can see I'm doing pretty interdisciplinary stuff. The way I got into this was funny. For my PhD, I was studying how the universe, space-time began. And there I had a couple of interesting discoveries in my grad school about how space-time began. And the findings I had were pretty wild. You've heard about some of these things in popular media, such as parallel universe and so on. Then there's quantum physics and relativity. And someday I asked the question, how come we are able to perceive things that we will never experience, nor can we experience, such as a black hole? Then I thought, wait a minute, is that a unique human ability to perceive things that we can never perceive directly? Abstract thought. Do only humans have abstract thought? And the standard question is, well, you ask the animals how to look at numbers, for example. That's supposed to be abstract thought. That sounds like an esoteric thing human beings have come up with. Is there any other evidence that animals perceive something abstract? And then the question came back from my quantum physics days to say, hey, space and time are two things which are abstract. Like if you were sitting next to me and I said, what is between you and me? And you'll say space. And we can't feel space. We can't smell it. We can't touch it. We can't hear it. But all those senses put together creates this perception of space. And not only is there a perception of space in human beings, every animal has a concept of space, and every animal agrees with each other on what that space is. probably the only thing the animals agree on, right? Zebras and lions will disagree on everything. Lions would think zebras are delicious and zebras probably won't agree, but they will agree on space. With millisecond and millimeter precision, animals agree on space, even though they are from different species. And it's abstract and they act on it and it's malleable. So I thought, hey, if I understand how our brain perceives space out of concrete things, maybe it'll give us an idea of how we perceive other things such as abstract thoughts and early universe and multiverse and mathematics and music and so on. I was still doing physics, and there I found out, hey, there's this part of the brain called hippocampus, which is known for like 50 years to be important for perception of space. And in fact, people have found that if you listen to one individual neuron from hippocampus, and there are like 500,000 to about a couple million neurons in human hippocampus. And if you listen to just one neuron in hippocampus as the subject is walking around, that neuron will fire or will be active at only one place in the environment, nowhere else. And a different neuron will be active somewhere different place, so on and so forth. And these neurons have a map of space, so-called cognitive map of space. And that discovery was made more than 35 so years ago, and it even got a Nobel Prize in 2014.
And these are what we now know as place cells?
Exactly. These are the place cells. And I said, wow, so now biologists have done amazing work for us. They have not only found which part of the brain is involved in perception of abstract space, but they also figured out what the neurons do. So there was only one little question I thought that was remaining. How? How do you go from these different visual and olfactory and auditory and other cues to neuron firing at one place? What is that place? And there was a lot of literature going on before that. You rotate the cues, you turn on the lights, you turn off the lights. The place cells do a lot of funky stuff, but it was hard to pin down how space arises. The literature got more interesting as I studied. Turns out if you have a subject sitting in a maze and the maze has certain walls, and if you rotate the walls around the subject, the many place cells, but not all, rotate correspondingly, suggesting the concept of place cell is driven by visual cues. Our cognitive map is based on visual cues, and that's the kind of the big cognitive map theory, which explains how place cells arise. But then the other weird things, this rotation of place cells with visual cues is true only for 30-40% of cells. Other 60% don't do anything. They remain where they are. What are they doing? Then story gets weirder. You turn off the lights. Place cells remain pretty much where they are. What happened there? Are they now having memory, or are they perceiving something else? Are they counting steps? How many steps left, right, and forward, so-called path integration? Or are they responding to some smells which are still there? Are they responding to some sounds?
Yeah.
The hippocampus will make map from everything, and how can we ever control it? So there came my so-called simple thought, what if we make virtual reality for rats? Because now in virtual reality, I can be very, very sure as to what stimulus tells a rat where he is. Smells, sounds, and textures will be there, but they will not tell him where he is in virtual space. Either the sound or the light or whatever else I put in will tell him where he is. So we thought, hey, now we'll be able to do a very clean experiment. We'll put the rat in virtual reality. It will be non-invasive. It'll be like the virtual reality used for humans, so that they are seated comfortably and the virtual screen is all around them, underneath their feet. Even they see the visual scene, they are fully immersed, unlike any other virtual reality system available to humans.
It's like the holodeck for rats.
Yeah, that's right. So we made virtual reality, and we thought, now we know that the only thing that tells the rat where he is are these visual cues that we put in, or the auditory cues that we can put in later if necessary. And then let's see what place cells do. That's where the study began. So we built that virtual reality. The first question was, will the rat buy into it? For us, we are so visual. Many people thought, and even we thought, rats are nocturnal creatures. They probably can't see well at all, and they'll focus entirely on the smell or texture, and they are seated somewhere and the scene goes around. They may not buy into it. Like many creatures don't care for television. So why would they care for this vision-based virtual reality? So the first question was, can they believe in virtual reality? Now, how am I going to to ask the rat, do you believe in it? That's a pretty hard one because we kind of speak different languages. So we thought, all right, here is one experiment we can do. In the virtual world, we will suspend virtual pillars. These pillars are suspended from the ceiling of the virtual world, and rat has to go underneath a pillar to get a little bit of sugared water. If he does it, then we'll know he's not only seeing the visual cues, he goes underneath them, which requires 3D perception of space. Within 3 days, the rat started to go underneath the pillar. In fact, we have videos on our website where if you put the rat on the very first day in virtual reality, he sees the pillar and he freezes. I don't know what thoughts went on, but I imagine he thought, what the hell is that coming at me? But in a matter of 2 minutes, he becomes very inquisitive, starts going towards the pillar, and as the pillar suspended above him, it's all virtual, starts to come closer, he starts to stand up on his hind leg to try to touch it. Totally, he buys into it. So then we thought, let's make it more difficult. So what we did is we made the virtual maze a little table about 2-meter diameter, suspended over the ground, just like in the real mazes that many people use. There is a little table on which the rat runs, and the table is above the ground, and the ground is the different visual textures such as tiles. Table has some random patterns such as matte or white and black and something. So we suspended the rat, let him walk on this virtual table chasing these pillars, and every now and then he'll reach the edge of the table. Now in the real world, with his whiskers and his eyes and his nose and his ears, he can tell if he took another step, he's going to fall. So he's not going to do that. What about the virtual world? Can he detect the edge of the world? So all we did is to say if he goes past the edge of the world, we simply stop updating the visual scenes. So we thought it's a tiny feedback, nothing major. Can he actually perceive the edge of a table? Purely visual. And believe it or not, in matter of 3 days, as soon as he sees the edge of the table, he takes a right turn, goes away. And in matter of 1 week, if he reaches the edge of the table, he gets curious every now and then, he goes up to the edge and then he does moonwalk. He walks backwards from the table. So he totally buys into it. Then we thought, okay, this is still the edge of the table. Let's make it really, really hard, which is the classic test of hippocampus, which is rat has to go to an unmarked location in that space. And if he goes to that unmarked location, he gets a bit of reward. And now there is nothing telling him where the reward is, except the triangulation with respect to all the landmarks on the walls. And it turns out rat can do that as well, as good as they will do it in the real world, where they could use all kinds of other cues such as smells or textures. Here it is nothing. It's only based on vision. They can go to an unmarked location very, very reliably, and they do it as well as the real world.
So in this case, the amount of data that they're getting from things like smells in the real world, it's sort of pouring water in a cup that's already full. It's more data than they even need.
Exactly. We thought, okay, so clearly it shows that the rats can see this stimuli in the virtual world. In every possible way, they can go to one particular stimulus to get a reward, they can see the edges, and they can localize an abstract space. So now we have 3 lines of evidence that they are seeing abstract space. That's pretty much all we could think of. These are classic tests everybody has done. So then we were super excited that, hey, these guys can see it. And there we did several additional experiments that started to then go kind of against what everybody expected. So first we thought, all right, let's now look inside the brain when he's doing this spatial task. And we thought, let's just be careful because this is virtual world. Who knows what's going on? So we made a real world maze which looks exactly like virtual maze in the next room. So both the mazes, real and virtual mazes, are in a dark room which is sound insulated. Both have same dark texture on the walls. Both have very similar smells. The virtual table is 2-meter diameter. The real room has an actual 2-meter diameter table. The walls have psychedelic or simple visual cues, and we can listen to the same neuron from the same rat in the real or virtual world. And then we can ask actually fascinating, potentially philosophical question: how does the brain tell apart what is real versus virtual? This is the question people have asked for the longest time. What is reality? And it has been not possible to do any scientific study in this way directly, because how do you mess with reality, which is convincing still? Well, here's virtual reality. It's allowing to do something where you can kind of tell it's not real, but it's compelling. So we listened to individual neuron. The first thing that happened is we found there was a neuron in the real world that was cooking away at very high rate. It was very active at a beautiful place field. You go to virtual world, this neuron neuron simply shut up, gone, not active at all. And we thought, well, that's weird. This neuron simply shut up. So we started looking more and more. And it turns out in virtual reality, compared to real world where everything as far as vision and rat's behavior are concerned are pretty much as similar as one can imagine. In virtual reality, 60% of hippocampal neurons shut down. That's a whopping shutdown. I haven't seen any other manipulation people have done of any kind, sensory stimuli, movement, some pharmacological manipulation, which will lead to 60% of hippocampus neurons simply shutting down.
Could that be related to some of the senses, like sense of smell, that weren't getting mapped?
Yeah, it could be. What we expected was it should be even better because the rat seems to be doing fine. So then we thought, all right, let's focus on the remaining 40%. So what do? And turns out these remaining 40% of neurons, they have no map of space at all. They are firing completely randomly. They have tiny amount of information, but pretty much at chance level. This opened up many questions. Why did this happen? And what is a place cell? Well, maybe if you look at the anatomy of hippocampus, which is known for like 100 years, unlike, let's say, visual cortex, which gets input pretty much from visual areas or eyes, auditory cortex, which largely gets input from the ears, and olfactory cortex gets input mostly from the nose. Hippocampus doesn't have that. Hippocampus actually is one part of the brain that gets input from pretty much every sensory cortex. Then should we expect hippocampus to make its mental map of space purely based on vision? Or will it also use smell, sound, textures, and so on? So that's our hypothesis, that maybe the way the hippocampus works is that it doesn't actually just rely on vision. In fact, if map had to be made purely based on vision, visual cortex can already do that. But our brain has to do a really difficult task to say this smell appeared at this place along with this sound, such as going by a pizzeria, you hear the sounds of pizzeria, you hear the fragrance of pizza, and you see the pizza shop board. If one of them was off, such as you had the sounds of a waterfall coming out of pizzeria, you will be freaked out saying, hey, something is wrong. And we take it for granted that all the different senses to us are always telling us consistent things. We take it for granted that, oh, that's what space is. But maybe hippocampus is actually putting these different things together to create that perception of space. And that perception of space is not just entirely vision. In fact, it needs everything. And what's going on in virtual reality is that when the rat is running on the ball has smells and textures and so on. They're just not telling him where he is, but in reality that information is coming through his nose and his whiskers and his legs and paws and so on. But they are not consistent with the visual cues. And that inconsistency is such a big deal that it totally wipes out the map and it shuts down 60% of neurons. So we thought, let's look at the activity of neurons in hippocampus in real world and virtual reality, not just as a function of where the rat is, but as a function of what the rat happens to be looking at that moment. So we did new experiments and analysis, and we found that actually the same visual cues which gave no spatial selectivity in hippocampus, they were actually able to modulate the neuron's activity as a function of which cue the rat was looking at, both in real world and virtual reality by the same amount. So now the story of place cells got even more funky. They were supposed to be abstract representation of space and independent of what specific thing the rat is looking at. Instead of that, we find that visual cues, which are supposed to give place cells, do not give place cells. So rather, visual cues alone are insufficient to generate spatial selectivity. But visual cues can directly influence hippocampal place cells. And we showed that actually if you make the visual cue wider or narrower, the neuron's activity becomes broader or narrower. And in fact, the entire hippocampal ensemble fires more when the rat is looking at that visual cue. But still, what about space? Is there any condition in which we can get some kind of spatial map in hippocampus in addition to just what he's looking at? So our hypothesis, as I mentioned, was that hippocampal neurons are creating map based on two sensory or motor modalities saying the same thing, at least two. So how can we test it? So what we did is we brought back those same pillars that rats liked, but we made just one little change. These pillars that are suspended in the air, they, instead of appearing at random places, appeared in only 2 places. So rat was going back and forth from pillar to pillar. And immediately spatial selectivity reappeared. 60% of neurons were still quiet, but the remaining neurons that were active, they immediately had place cells, but they were actually funky place cells, in the sense that these neurons were active at the same distance from the pillar, regardless of where the rat was in that space. So these neurons with vision, they were actually not counting the rat's absolute spatial location. They were counting relative distances. And that says that, hey, these neurons potentially were keeping track of, hey, that pillar is going to give me reward. How far am I from the reward? And that starts to explain many other findings people found long time ago, such as when you turn off the lights, place cells remain in the same place because place cells are responding to at least 2 sensory modalities. And in reality, they are doing some multi-sensory representation. So it sounds abstract from the point of view of pure vision, but it's actually not abstract. It's some combination of things.
Sounds like you need something like a quorum among your neurons. You need a certain number of votes to say, yeah, I definitively am where I think I am.
Exactly right. So that starts to have many implications for how hippocampus works, what place cells are, and what might be going on in some diseases. Alzheimer's disease starts in hippocampus. One of the most common form of epilepsy that is intractable, no drugs can cure it, is in hippocampus. PTSD, schizophrenia, depression, hippocampus is involved. And in fact, Alzheimer's patients tend to get lost in space. And the converse is also true, that people who have phenomenal memory, such as memory athletes, if you ask them, how do you remember lots of places? They use a classic method called method of loci. They create a mental memory landscape, a big landscape with buildings and shops and so on. And every card that these memory athletes are asked to remember, they assign each card to a certain building in that landscape, and then they simply walk through the landscape and read off the cards. So there is something fundamental about space and memory. Memory about things which is not just space, but even things such as conversation. So this whole thing starts to say something about how memories themselves are formed. We think memory is one thing that we recall or don't recall. But in reality, memories too are some multisensory combinations. And if hippocampal neurons are shutting down in virtual reality like this, and they have such abnormal responses, no spatial map left, you only wonder what will be the effect of these abnormal responses on hippocampus itself in the long run. Because as we know, neurons that fire together wire together. That means that if neurons misfire together, they might miswire together. We have not found any evidence of the long-term effects of using virtual reality, either positive or negative. We simply haven't completed those studies, but I believe it's quite crucial to study this.
Yeah, especially as people are conceivably going to be spending more and more percentage of their time in virtual reality as the technology improves.
That's exactly right. So we showed that if a rat simply walked in a room back and forth, just a 2-meter long track, spent only 20 to 30 seconds, the entire hippocampal map changed by 2-fold. They were not just very young rats, they were actually adult rats. So we have to think carefully as to what will happen with virtual reality use for prolonged period to adults and children or youngsters or young adults.
Part of me also wonders, is there a flip side, like the way that Olympic athletes will train at higher elevations where there's less oxygen, because that actually strengthens their body, and when they go back to a higher oxygen environment, they're better as a result of it. Might there be the potential for almost doing like memory training in a VR environment, and then when you come back to the real world, you've got memory superpowers?
These are great questions. The bottom line is we don't know whatever is happening in virtual reality, does it have any long-term consequences at all, good or bad? You see, hippocampal neurons are doing all kinds of things. Vast majority of it we actually cannot perceive. So I give you an example. If you focus on again the hippocampus, and if you looked at not just 1 or 2 neurons, but millions of neurons all at the same time, what you notice is something fascinating, which is that in addition to these neurons firing at certain place, the entire hippocampal activity becomes rhythmic, and it develops about an 8 hertz rhythm, and I can even tap it out. That's roughly the rhythm. You can play it by your fingertips. As soon as subjects, they start to walk, this rhythm appears. As soon as they stop, the rhythm is gone. As soon as they fall asleep, the rhythm is still gone. And when they are deeply asleep and then they go into REM sleep, the rhythm is back. So this rhythm called theta rhythm, which is about 10 times a second, that rhythm is there only when the rats are running and when they are dreaming. And what we found is place cells typically are active for about 1 second. That is not enough for neurons to wire together. What is necessary is that two neurons have to fire much closer together. And what this theta rhythm does is it kind of compresses that behavioral sequences and make sure the neurons, whatever they are doing on the scale of 1 second, they are doing the same thing on the scale of 1 millisecond. And that is how they wire together and how they remember things.
Almost like a metronome at a cellular level?
Exactly. It's a metronome at a cellular level, but the metronome is super fast. Within one theta cycle, which is 10 times a second, you'll have 10 or 20 additional metronomes going on. As the rat runs faster in virtual reality, the theta rhythm doesn't speed up. So the rhythm itself has changed in a fundamental way. But the neuron's language with respect to the rhythm, that language is still the same. The place cells are gone, but the metronome language is still as good as the real world. According to our hypothesis now, based on the data, perhaps these neurons are not just keeping track of space. What they are doing is keeping actually track of brief segments of time. And these brief segments of time are stitched together using this metronome to create a perception of space. So your hippocampus is doing a really intricate calculation. And as you are walking in the real world, everything is happening. So you can decide whether to run from the lion or go towards a tree and get food. And these are massively complicated calculations, and in virtual reality, these laws are being broken. Never before in history of animals, two senses said one thing is happening and two other senses said, nope, that's not happening. Long time ago, a primitive animal sitting inside its cave and looking outside and watching the giraffes go by. In that case, all the senses are saying the same thing. The giraffe is walking, you are not walking. That's not true in virtual reality. Some senses say you are walking, and as a result, the world is changing. Some other senses say, nope, that's not happening. You're not walking because nothing is changing.
We've got the primary five senses that we always think about, the sight, hearing, smell, touch, and sense of taste. And those get so drummed into us, I feel like it's easy to forget some of the very, very important secondary senses, things like proprioception of where is my body physically existing in space right now. That's nowhere on our primary five, but obviously very important.
Exactly. And that's fascinating because now this research in virtual reality reveals that not only that's an important sense, but that important sense is driving these memory-making neurons directly.
This reminds me of something called dead reckoning, that when sailors in the old days were trying to figure out where they were at sea before they had GPSs and things like that, ideally you could see the stars at night and that would give you a way of triangulating your position. But if it was a cloudy night, kind of the best they could do for a long time before they had accurate time measurement was say, well, we've been going east and we think we've been going at this many knots, and just doing the multiplication. And it sounds like at least some portion of our brain is kind of doing that. Like, well, I've been walking at this speed for a certain amount of time, so based on the last compass reading of my position looking at physical objects, here's where I must be.
That's right. And we are seeing evidence of that in single neurons in hippocampus. So virtual reality allowed us to uncover this calculation that neurons are doing. In the real world, of course, it's hard to see this because as soon as you take 2 steps, the olfactory stimuli or the tactile stimuli, the gravel on the ground says, hey, forget 2 steps, you are on a slope, which is far more important than how many steps you walked. And they are also unmasking another thing. In the real world, people have noticed for the longest time since place cells were discovered that these neurons, whenever they are active for like a whole second. Each place field is like 30-40 centimeter long. Why so broad? Why not 2 centimeter? Because I should be able to tell where I am with respect to my table, not with an accuracy of 40 centimeters or half a meter. I'll just hit everything. I should be able to tell cognitively with an accuracy of a couple of centimeters. Then why are place fields so huge? Is that a bad thing? Well, it turns out in virtual reality, even though place cells have no map, they are still active for 1 or 2 seconds. Even though they have no spatial map, their metronome map is still intact. What happened there? So what we think is going on is that the body or the brain has that information that I walk forward for 5 steps. And if my brain had to recompute at every millisecond where I was, it'll be very expensive. Based on evolution, the brain has sort of figured out the basic law of physics that the world that we live in is continuous, barring few exceptions, which means that if I took a step forward, then most of the time, about 99.99% of the time, the world is going to be exactly the same for that 1 second. So instead of recomputing my steps every millisecond or every centimeter, the hippocampal neurons are going to say, you know what, for that 1 second, I'm going to cruise along and say I'm in the same place. And then use that information that I went that one block of memory or one block of space, which is metronomed with that theta rhythm. I'm going to use these chunks to build space.
That's really fascinating because it's almost like our brains are pixelizing physical space and turning it into these discrete chunks that we think about as being more like an artifact of the way that computers work. I wonder to what extent some of these same mechanisms within the brain might be used to keep track of where, let's say, my right arm is in space at any one time.
Yeah, so that actually brings two important questions. So one is space with respect to ourselves, and other is what happens in rats versus humans. So let's answer both questions. So far, the belief has been that distances with respect to the body, they are kept track of by other brain regions. For example, the parietal cortex. Parietal cortex is one major sensory cortex that sends information to hippocampus, and hippocampus uses all that information to create this other space, which is not just around me, but far away from me. The second question, are these results at all relevant for humans, or is kind of worthless? So then we studied some studies based on humans. How do people measure signal from human brains? Well, as I mentioned, hippocampus has many neurological diseases rooted in it. One of them is intractable epilepsy. And when some patients have these intractable epilepsy, they have to find out where the epileptic focus is. And in order to find that epileptic focus and remove that tissue, that's the only way to cure this epilepsy. What people do is to operate on that human being, insert electrode in hippocampus and wait for an epileptic seizures to say where the seizure started and detect where the bad tissue was and then later on remove it. And while the patients are waiting, some group of scientists let the patients navigate in a virtual world. Of course, not as immersive as our virtual world. It's just a monitor in front of the patient in a hospital room. But what they found there too is that as long as the patient was navigating a room randomly, like the rats are walking randomly, there was no or very little spatial selectivity in place cells, even in humans, in virtual reality. Now, of course, they can do head-to-head comparison to say, okay, the patient runs in the real world versus virtual world. Did the neurons shut down? Were the place cells intact in the real world? And so on. Those things cannot be done yet because these are patients and you can't ask them to walk around. And the concerns remain that patients had epilepsy. So what you see in the brain could be because of epilepsy and not other reasons. But at the same time, the results based what we see in rodents, these neurons did behave like place cells in real world, but they actually shut down and did not behave like places in virtual reality. It is due to something about virtual reality, not because it is not compelling, not because they are rats. It is something more fundamental.
How close are we conceivably to creating a rat matrix, really raising an animal in a virtual environment so we could kind of see if that was the baseline, how its brain would behave?
Yeah, fascinating questions. In fact, the question that we want to ask is if a rat experiences virtual reality several hours a day for 3 months to 4 months, what happens when that rat goes to the real world? Because in rat's life, 3 to 4 months is like 20 years in human life.
Right.
And we can get an idea of what are the effects of virtual reality experience, at least on human adults. If we were to strap on virtual reality goggles for an hour a day for a couple of years, what will happen to our perception in the real world? In fact, as you see, hippocampus, what happens during behavior resurfaces during sleep. Will virtual reality even influence their dreams? We don't know. These are things to do.
Which of these strands would you like to pick up next in your research? Where are the biggest nagging questions for you at the moment?
The first question we want to know is what is that space? That's what we started with. Virtual reality was supposed to be magic bullet. They'll give us simple answers. Instead of that, it uncovered all kinds of funny things, such as distances and times and metronomes. So we want to now start putting these things together to see, okay, what is reality? And perhaps start asking that interesting but dangerous because it's a difficult question. attention as to what is real, what is unreal. How is the brain reacting so differently? We have some hooks into it. We want to pursue this further because already we have uncovered it responds to locomotion, to internal rhythm, to some metronome, to smell, sound, texture. That's a very complicated arithmetic these neurons are doing. So we want to figure out how it responds to each one and then how it puts together. If we can do that, we will go pretty far in understanding how we create abstract perceptions. That'll be fun.
I must admit, part of me feels kind of nice that there's some primacy still to physical reality, that we haven't quite cracked the nut of faking it yet.
Yeah.
The sort of conservative old school of like, there's something ephemerally better about reality that technology hasn't broken yet.
I don't want to provide the message that virtual reality is all bad. We want to be careful, that's about it. But I also dream, when we started these experiments, neither us nor anybody else ever imagined that in virtual reality, 60% of neurons will shut down. That's a wild thing. But when something like that happens, that raises hope that, hey, what if we can now create virtual reality of suitable kind, and we are working on it, where we can use virtual reality to either shut down hippocampus or make it active or make it super active. Maybe make it active more than normal reality. I think it should be possible.
It's fascinating to think about what perceptually that might feel like if you're able to sort of overclock the activity of the hippocampus. Would that feel more real than reality?
Yeah, it's a win-win. We can get an idea of what reality is. And if we can do it, we can start having virtual reality treatments for these diseases. Because if you look at epilepsy, what's the problem with epilepsy? High amount of neural activity. What's the problem with Alzheimer's disease? Too few neurons active. Maybe we can use virtual reality to rewire hippocampus to start developing therapies for some diseases for which there are no pharmaceutical treatments available yet. And there is some hint that that may be possible.
Smart Drug Smarts.
So a very big thank you to Dr. Mayank Mehta for taking the time for that interview. This seems like an incredibly cool area of study for me. If I were in university now and looking for something about the brain to study, this would not be a bad area to specialize, both because I think we can learn something fundamentally interesting about how we come to feel that we're in a space, kind of that whole mind-body problem. And there's also just bound to be a ton of technical implementation questions as more and more aspects of our lives start migrating into these virtual environments, which I think is not very debatable that we're going to be living more of our lives in virtual environments, working to figure out how to make our brains feel neurologically at home there. There's potential for both the best and worst of science fiction style outcomes, but I guess one of the questions that we don't think about a lot is how do we protect our brains as we do so? Is there a need to keep reality from tipping its hand, having a couple of our physical senses that are out of sync with the data coming in from our VR modulated senses? If I'm seeing and hearing some pleasure palace, does it really matter if I'm smelling my dirty laundry in the next room? Maybe it doesn't and it's no big deal and we can just tune that out. Or maybe having sensory asynchronies like that is actually something that's fundamentally bad in a way that we don't understand yet. And spending too long in that state, we could kind of lose our moorings on, yes, this is the real reality that I'm in. Who knows, but unanswered questions and definitely a very interesting topic. And we'll keep somewhat on the theme of navigation through physical space, and in this case, how it relates to anxiety in the Ruthless Listener Retention Gimmick.
Smart Drug Smarts, Ruthless Listener Retention Gimmick.
So I'm going to wager a guess that not very many of you know the word etiology, which I did not know. I had to look this one up. It is a great way of getting rid of some extra vowels if you're playing Scrabble. The British spelling actually has 4 of the first 5 letters as vowels, A-E-T-I-O-L-O-G-Y. But what etiology is the branch of knowledge specifically concerned with causes, and even more specifically, the causes and origins of disease. So etiology could be, why do I have the sniffles, or which of my girlfriends gave me gonorrhea? But researchers at King's College London were looking at the etiology of anxiety some people feel when faced with math or navigational problems. Nervousness about a math test or having to parallel park. And could some of these feelings be traced back reliably to a person's genes? They looked at a very large database of more than 1,400 twin pairs aged 19 to 21. These twins had been questioned about several different forms of anxiety: general anxiety, mathematical anxiety, and spatial anxiety, broken down in 2 ways, one related to navigation through physical space, and the other related to rotation and visualization of objects. Can you picture what an elephant looks like flipped upside down and twisted counterclockwise, that kind of thing. Among other things, the study showed that a lot of these types of anxiety are dissociable. Just because somebody's nervous about things like parallel parking doesn't necessarily mean that they're going to be nervous about playing Jenga or doing a jigsaw puzzle. One thing that was consistent was that DNA explained a lot of these characteristics, with over a third of the differences between people in their levels of anxiety in these areas being traceable back to their genes. Non-shared environments were found to explain the rest of the differences, non-shared environments being things like people's experience with driving, cycling, or playing 3D computer games where they're moving through a virtual space. Spatialization skills, which as you might imagine are inversely correlated with spatial anxiety, have been linked to success in STEM professions, science, technology, engineering, and mathematics. And so the researchers were curious just how much of a barrier a person's DNA might be if they were on the lower end of this skill spectrum. Said researcher Margherita Malancini, pinpointing specific genes for anxiety could help in identifying children who are most at risk and subsequently to intervene and prevent the development of anxiety in these different contexts. For children at a greater genetic risk of mathematics anxiety, interventions aimed at enhancing motivation and providing positive feedback may help with increasing performance in this area.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, so you heard it. That was all for episode number 191 of Smart Drug Smarts. Thank you for hanging around with us until the very end. If you're curious about anything that we talked about here, everything will be up online at smartdrugsmarts.com/191. If you missed last week's episode in 190, I spoke with Dr. Jed Fahey about the compound sulforaphane, found in broccoli sprouts, cruciferous vegetables, and a few other places. Also an African tree called moringa that we'll be talking about in a coming episode. And next week in 192, I'm not exactly sure what we're going to be covering. There's a couple of viable contenders. And the week after that, we've got Left-Handers Day coming up, so we may try to do something special for all of you Southpaws out there. Also have a delectable overdose edition coming at you soon. But either way, I will see you back here next Friday. Same time, same podcast, and with that same unwavering commitment to helping you fine-tune the performance of your own brain. Have a great week, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.